My name is Joshua Mugabi. I welcome you to Topic 1 Cell Biology. Our learning outcome A. Analyze the properties and functions of chemical compounds, looking at water, lipids, proteins, including enzymes from mammals in a cell, focusing on their roles in maintaining cellular structure and metabolic processes in living organisms. We are going to look at learning activity B. Using modeling or dramatization, learners discuss the mechanism of enzyme action. We are going to look at lock and key, induced-fit hypothesis, and we shall look at the different types of enzyme inhibition, and then make a write-up and present it to the class in a plenary. Let's start with the lock and key and induced fit hypothesis. Item 1. We shall start with enzyme specificity in digestion. During a digestion experiment, students add amylase to starch and observe a rapid breakdown to maltose. When amylase is added to protein, no reaction occurs. One learner suggests that the enzymes work like a lock and key, while others argue that the enzyme changes shape slightly during the reaction. Task. Explain why amylase acts on starch but not on protein. Describe the lock and key hypothesis of enzyme action. Describe the induced fit hypothesis of enzyme action. State one advantage of the induced fit hypothesis over the lock and key hypothesis. Explain why amylase acts on starch but not on protein. Amylase is specific to starch because its active site is complementary in shape to the starch molecule. Protein molecules do not fit the active site of amylase, so no enzyme substrate complex forms. Describe the lock and key hypothesis of enzyme action. Lock and key hypothesis. The enzyme has a rigged active site. The substrate fits exactly into the active site. An enzyme substrate complex is formed. Products are released, leaving the enzyme unchanged. Describe the induced fit hypothesis of enzyme action. Induced fit hypothesis. The active site is flexible, not rigid. When the substrate approaches, the enzyme changes shape slightly. This brings the substrate into the correct position for reaction. The enzyme substrate complex lowers activation energy. State one advantage of induced-fit hypothesis over lock and key hypothesis. The induced-fit hypothesis explains how enzymes can bind substrates more precisely and stabilizes the transition state, making it more accurate than the lock and the key model. Still under lock and key and induced-fit hypothesis, item 2, we are going to look at the effect of temperature on enzyme action. Milk containing lactose is treated with the enzyme lactase at different temperatures. At very high temperatures, lactose is not broken down. Task: explain why enzyme activity decreases at high temperature. Relate this observation to the induced-fit model. Explain why enzyme activity decreases at high temperature. High temperatures cause denaturation of enzyme. Bonds maintain the enzyme's tertiary structure break. The active site changes shape and cannot bind the substrate. Relate this observation to the induced-fit model. Induced-fit requires a specific enzyme shape. Denaturation prevents the enzyme from changing shape appropriately to bind the substrate. Now let us look at different types of enzyme inhibition. Item 3. We shall look at poisoning case in a laboratory. A pesticide accidentally contaminates a water source. Victims show symptoms of nerve malfunction. Scientists discover that the pesticide prevents enzymes from functioning. Task: define enzyme inhibition. Describe competitive inhibition. Describe non-competitive inhibition. Explain why some poisons are difficult to reverse. Define enzyme inhibition. Enzyme inhibition is the reduction or prevention of enzyme activity by an inhibitor. Describe competitive inhibition. Competitive inhibition. The inhibitor has a similar shape to the substrate. It competes with the substrate for the active site. Increasing substrate concentration reduces the effect of inhibition. Example, malonate inhibiting succinate dehydrogenase. Describe non-competitive inhibition. Non-competitive inhibition. The inhibitor binds to a site other than the active site. It alters the enzyme's shape. Increasing substrate concentration does not overcome inhibition. Often leads to permanent enzyme inactivation. Explain why some poisons are difficult to reverse. Some poisons cause irreversible inhibition. They permanently alter the enzyme structure. Normal enzyme activity cannot be restored. Still under different types of enzyme inhibition, item 4. Let's now look at drug use in medical treatment. A patient is prescribed a drug that slows down an enzyme involved in cholesterol synthesis without completely stopping its action. Task: identify the type of inhibition involved. Explain how this type of inhibition is beneficial in medicine. Suggest why dosage must be carefully controlled. Identify the type of inhibition involved. Competitive inhibition. Explain how this type of inhibition is beneficial in medicine. It reduces enzyme activity without stopping it completely. Normal metabolic balance is maintained. Suggest why dosage must be carefully controlled. Excessive inhibitor may prevent normal enzyme function. Insufficient inhibitor may be ineffective. Examiner key points. Use correct terminology, active site, substrate enzyme complex. Explain processes in logical steps. Distinguish clearly between types of inhibition. Apply knowledge to real life situation. Thank you for participating in topic 1, cell biology. Our learning outcome A, analyze the properties and functions of chemical compounds, water, lipids, proteins, including enzymes from mammals, in a cell, focusing on their roles in maintaining cellular structure and metabolic processes in living organisms. We have been looking at learning activity B. Using modeling or dramatization, learners discuss 1. The mechanism of enzyme action, that is lock and key and induced-fit hypothesis. The different types of enzyme inhibition. Then you make a write-up and present it to the class in the plenary. If this has been very useful to you, please visit me at Mugabi Biology on YouTube.